in order for a protein to have a particular function, they have to fall into a specific structure. And that is determined by the amino acid sequence. So that's why predicting the structure from the sequence is important. Because if you have the structure, maybe we can understand the function. And also, in my opinion, this is the most important problem in molecular biology. That is, genotype determines phenotype, right? Genotype means the DNA sequence as a whole in our cell. And that information determines how we look or how we work or function. What's interesting about this scheme is that genotypes are essentially information. They are digital information. So they can be expressed as a string of letters. A, T, G, C, right? But phenotypes are very diverse. We have fingers, we have eyes, we have brains, and many things. And we can do many things. All those things are determined, to some extent, determined by a genotype. So how genotypes can specify this phenotype is the most fundamental problem in molecular biology, in my opinion. And why proteins are important is here. Proteins are direct copies of DNA, more or less. And it can also form a structure and function. So protein is at the pivotal place in this flow of information to material. That's why how protein molecules fall into a specific structure is important. So the problem of protein structure prediction is, given a protein sequence here, which is made of 20 types of amino acids, predict the native structure of the protein. The problem is very simple. I mean, stating the problem is simple, but solving it is very, very difficult. Actually, this problem has been proposed more than, I guess, 50 years ago, but it hasn't been solved completely yet. So how can we attack this problem? First, after all, proteins are molecules, nothing but molecules. So let us represent the molecule like this, bunch of atoms. And if you have molecules, you have atomic nuclei and electrons. So maybe we can set up a Schrodinger's equation. Just solve it with a particular boundary conditions and some other conditions, like temperature, water concentration, etc. But solving this equation for this big molecule is hopeless. Too much computation is required. So I bet you cannot finish solving this equation until, well, maybe after you die. So we have to give up this. So as an approximation, we kind of approximate this quantum system into a classical system so that we can solve Newton's equation instead. But to solve Newton's equation, we have to have some kind of potential function, which are ultimately determined by this. But we don't know the precise mechanism of these potential functions, so we have very, very rough approximation. Inevitably, these approximations are all usually very inaccurate. So even if we use lots of supercomputers and lots of CPU times, the global energy minimum conformation may not correspond to the native structure. That's because the force field is inaccurate. So that leads us to consider some other alternative approaches. And here I introduce some empirical or statistical methods based on database. Here's a time series of the number of entries in protein data bank. It started in 1971, and it's growing like this. So now we have more than 11,000 entries in the protein data bank. So using this kind of information, we can statistically estimate correspondence between the sequence and the structure. But explaining three-dimensional structure prediction is very complicated because three-dimensional structures are complicated. So in this talk, I concentrate on a simpler problem called secondary structure prediction. If you represent protein structure like this, you can see helix here and some extended strands here, right? So these are called secondary structures of proteins. And if you represent this helix as a cylinder and these strands as arrows, the yellow arrows here, you can represent the sequence and the secondary structure like this. So instead of predicting the three-dimensional coordinates, secondary structure prediction predicts a second string of secondary structures like this along the sequence. So it's a lot easier problem. Of course, in my research, I studied three-dimensional structure prediction problem, but for the sake of simple explanation, I explained this secondary structure prediction. So how can we predict the secondary structure from the sequence? Here's the basic idea. If you have a sequence, amino acid sequence here, take a local fragment of a sequence, for example, 5 residue fragments here, and feed this information of fragment into some function which outputs one of the 3 states, helix or extended strands or others, coils. So this is 3-state prediction. This function can be anything. The simplest one is linear regression function. It's very easy. Or it can be more complicated using nonlinear functions like neural nets or support vector machines or anything. And here's a simple results of secondary structure prediction. If you use linear regression method, which is the simplest possible method you can think of, the accuracy of secondary structure prediction is something like 65%, which is good, but not too good. And if you use a little more complicated method using machine learning, like neural networks, this accuracy increases by 2%. That's moderate, but still a little better. But how can you improve these results? You know, this is okay, but we want more accurate results. So one important ingredient in this field is the use of evolutionary information. If you have many protein sequences, you can draw this kind of dendrogram or evolutionary tree. So since many diverse organisms came from, it is assumed to come from a single species. As evolution proceeds, many variants of many proteins have emerged. So we have some evolutionary relationship between proteins. So using this kind of information, we may improve the protein structure prediction problem. And the reason behind this is if you have similar sequences, these proteins have similar structures. So if you align 2 sequences like this, and these 2 sequences have 50% identical amino acid residues here, and if you superimpose the structures, they're essentially the same, right? So if you put together the information of many related sequences, maybe that will enhance the information of the sequence. So this is the motivation behind the use of evolutionary information. So let us collect many sequences, as many sequences as possible, of a target protein of your interest. So here's a multiple sequence alignment of a particular protein called myoglobin, or globins. there are many, many sequences, and you may get dizzy. And maybe you don't see any patterns, but if you look carefully, there are some patterns. I cannot see it from here. Where is it? In some places, there are hydrophobic residues dominant. And in some places, there are many prolines here. And let's see. But anyway, it's too hard to see. We don't read it by our eyes. We use computer programs. So by taking the statistics of each column here, where each row is a sequence, and each column corresponds to a site in the protein structure. So we can take the statistics of each column, and this is the site along the sequence, and these are 20 kinds of amino acid residues. And in this position, some particular amino acid called tryptophan is very dominant. And in this place and this place, some other amino acid called histidine are dominant here. In this way, we can summarize the information, evolutionary information of many protein sequences. And these can be used as an input to the previous function. So how does this improve the prediction? The first two rows are the same as before. And if we use PSSM, by the way, this is called PSSM, position-specific scoring matrix. This is position-specific. So if you use PSSM with linear regression, now the accuracy is 75%. This is 10% increase from the previous result. And if you combine PSSM with machine learning, now the accuracy is 80%. And if you compare this, previously, if you use only a single sequence, moving from linear regression to machine learning improves only 3%. But if you use PSSM with linear regression, this improves 10%. And if you combine that with machine learning, the increase is what? 15%. And interestingly, if you use only single sequences, this is 3%, right? But if you use PSSM, this increases 5%. So if you use richer information, then machine learning becomes more effective. So how should we interpret this result? First of all, proteins are molecules, so they are physical materials. And in the real-time scale of a single organism, protein folds from a random coil to the native structure. This happens in the order of seconds or milliseconds. And if you consider the evolution of proteins, this is the time scale of more than 1 million years, maybe billion years. And if you use this information, you can summarize the sequence in this way. So, to combine these two facets of protein structure, you can draw this kind of diagram from a single sequence. By the laws of physics, it folds into the native structure. This is thermodynamic equilibrium. But in the course of evolution, protein structure is conserved because it must conserve the function. If the function is lost, then an organism dies. So it must evolve in the way that the essential function is preserved. And that conservation creates the pattern of the sequence. So in this sense, the protein structure actually determines the protein sequence. So here's a cyclic logic in protein structure prediction. So, in the summary, so the essence of protein structure prediction is to understand how this cycle become consistent with each other. You know, physics and evolution, they may look completely unrelated, but they are deeply related. So physics and evolution must be integrated to understand protein structures. And what this means is that we usually think physics is more fundamental than biology. So biological phenomena should be understood in terms of physics. But to start with, our understanding of detailed interactions and physical interactions were imprecise. So that's why we resulted into the use of evolutionary information. But using the evolutionary information, maybe we can understand the physics deeper, more deeply. So in this sense, biology can be more fundamental than physics in this problem setting. And I think this is an interesting viewpoint to consider. Okay, that's all. Thank you.
Thank you, Professor. Yeah, of course, like our body is built up of proteins, and most of the diseases are some effects of the protein. If you cannot predict the structure of protein, you cannot propose a new drug. In this sense, the structure prediction is really important for the future evolution of several medicines. And now the session is open for the discussion, and we have 3 or 4 minutes for the discussion. Please raise your hand for the to start the discussion, please.
Okay, it is very simple to predict the secondary, the structure through the sequence of amino acid like alpha helix or beta sheet. But sometimes it is very, quite difficult to predict the, the tertiary conformation. But I think if you use the X-ray diffraction method, crystallization, the protein, then you can precisely predict, or not predict, precisely you can determine the conformation of the protein, rather than the theoretical one. But because the theoretical, this conformation is more deviant from the, the original conformation. Sometimes, not sometimes, most of the cases. We also mentioned that the accuracy is around maybe 70% or 60%, 65%. This is the one thing. So, definitely, the research is, the interest, growing day by day on the, the protein conformation and PTM. But the another thing is that the protein is not all the time is, the conserved, because if the point mutation is occur in a very active site, then it's become the malfunction of that protein.
That's true.
Yeah. So I think rather than going through the X-ray diffraction method, when it is it's not possible to predict the conformation only based on the sequence only.
Well, my point here is I'm not really interested in practical aspect of this problem. I'm interested in more in the fundamental problem of biology. So I want to understand the correspondence between the genotype and phenotype. And that's why this problem itself is important, apart from its practical importance. As for practical aspect, actually nowadays the most efficient way to predict the protein structure is to combine with experiments.
Yeah.
So if you know the protein structure for some protein, and if you have a target protein to be whose structure to be predicted, and if the amino acid sequences of these proteins are similar, then you can guess if they have similar structures. And this is the most reliable way to predict the structure right now. Yes.
I have a question, please.
Yes, please.
First of all, thank you very much. It was really an interesting talk. So So my question is that, for example, if we have similar sequences, and definitely many tools are available for predicting the 3D structure, and recently I am using iTaser. So what is your suggestion that iTaser is good, because different papers and different literature predict different kinds of, different literature suggests different kinds of way. But what do you think, sir? iTaser is good for predicting the structure, or maybe you can suggest some other?
I cannot suggest any other, but ITASA is fairly good. But you have to be very, very careful because it can fail very often.
Oh, you mean, sir, I have
No
to be
method is good enough.
Oh, so you mean, sir, I have to be careful for refining the structure?
Right.
Okay, thank you
So
very
just
much.
don't trust that prediction.
Okay, thank you very much.
You can accept one short question.
One short question.
Okay, from the back side.
Thank you very much. So just my question is not about science, it will be little about philosophy. So why in physics we can move forward for quantum physics, but we biologists, we are blocked. We cannot move in quantum biology. So if I express myself like in easy words, so the Schrödinger cat, they could find enough scientific tools to prove that the cat, if it is in the cage, whether it is dead or alive or it is in between. Now the physics can make it like it's the Schrödinger equation. But in biology, is it a lack of philosophy, imagination, or is it a lack of experimental tools?
I don't get your point. What's your question?
So my question is about quantum biology.
Yeah, quantum biology. Yes, yeah, quantum biology.
Yes, but in quantum biology we cannot make progress same as in physics. The progress in quantum biology
Well,
is...
I think most of the cases, the problem is the complexity of biological systems. There are too many atoms. But in ordinary physics, they just deal with a small number of atoms. Or in case they treat large number of molecules, they only treat homogeneous system. But biological systems are just heterogeneous and very complex. So it's hard to deal with.
So that means it is lack of scientific tools.
Yeah, that's one reason.
Thank you very much.
So you may proceed the discussion in the break session, please. And join me to thank again for the professor for the great talk. And moving to the next session, the invited talk 5, let me invite Mr. On Wahyudi from Shizuoka University. And he's going to talk in functional analysis of TI lipocalin proteins in tomato.
Assalamualaikum warahmatullahi wabarakatuh. So today I would like to talk about the functional analysis of lipocalin protein in tomato. So actually I'm worried about today because I present after Professor Akira. So Professor Akira like the top building, but for me like the first floor of the building. So my knowledge is not enough. So, but today I would like to talk about the applied science about the protein. So, in our laboratory in Shizuoka University, so we study about the TIL lipocalin protein. So we use microtome. So we got that a tomato, this tomato is very unique tomato. So we got from Professor Azura in Tsukuba University. So this tomato is filial number 12, so breeding 12 filial. So very unique because not so high, so only 20 centimeters. And then dwarf tomato, and then very easy to growing up in the under fluorescent light. So we can do this in the laboratory, and also we can develop easy in the soil media and using the hydroponic media. So this, for these reasons, so we use microtome for our project or our object of genomic study. So our laboratory we study to understand the plastic differentiation. During that study, so we want to understand that mechanism of differentiation from chloroplast to chromoplast. So using 2D patch, that means two-dimensional polyacrylamide gel electrophoresis, we detected lipocalin protein, TIL, temperature induced lipocalin, in the different type of tomato. In the red tomato, actually we detected two TIL, even though in the PI is different, and then the number of TIL spot is different. So we thought that TIL are related in the fruit colors and the differentiation of chromoplast. And then I want to introduce about what is lipocalin protein. So lipocalin protein actually a group of protein. So widely it's distributed in animals and then bacteria, but very little known about plant lipocalin. Thank you. And then plant lipocalin actually divided into three families. So first is TIL, temperature induced lipocalin, and then chloroplastic lipocalin, and then lipocalin-like protein. But today I would like to talk about only TIL, TIL1 and TIL2 lipocalin protein. This protein have function, first is environmental stress response. And then also this protein have function to induction apoptosis, and then membrane formation, and then regulation of immune response, and then cell growth and metabolism adjustment. Many researchers study about this protein and focus on the environmental stress response. So TIL in every plant have function to abiotic stress, like heat tolerance, low cold tolerance, and then drought stress, salt salinity, osmotic stress, and others. So that information was published in the several journals. And then to better understand the relationship between fruit color and then chromoplast differentiation and lipocalin, so we were performing the following experiment. So our protein, TIL, actually identical with other lipocalin protein TILs. So in the tomato TIL, SLTIL means Solanum lycopersicum TIL, so tomato TIL, actually identical 75 until 96% with other TIL, like Medicago, and then Nicotiana tabacum, and then Solanum lycopersicum, Solanum tuberosum, and others. But most identical tomato TIL with the Solanum tuberosum, or potato. And then TIL2 also, our sequence, and then check using the bioinformatic and CDI. So our protein identical 96% with Solanum tuberosum. And then we setting experiment. So where is the location of our protein in the cell? So we want to check, and then we designed an experiment. So we use PUGW5 vector and then PUGW6 vector. And this vector is N-terminus. So we can fuse frame that 35S promoter and then our target gene, TIL1 and TIL2. And then using GFP fusion, we can check the N-terminus, the localization of our protein in the cell. And also 35S promoter, GFP and TIL1, TIL2, we can use using C-terminus in the fuse in the plant. So we transform that construct using a particle bombardment. Using PDS 1000, we can understand where is the localization of this protein in the cell.